So hi everyone, welcome and thank you for coming to today's Thursday seminar. Today's talk is actually the last of 2020 and will be a little bit different than our usual Thursday seminars because it's going to be given by Shai Ben David. So Shai is a professor of computer science at the University of Waterloo, and he is also a member of Vector's faculty. So I'll hand over to you, Shai, and let you get going.
Okay, hi. So welcome everybody. So yeah, what I want to talk about is the statistical significance of the COVID vaccine testing results. And I think I have two goals in this talk. I mean, one of them is, of course, to clarify to what extent you can trust the efficacy of those vaccines based on the data that we know. But the other thing is I just want to demonstrate how we can use very simple statistic tools in order to analyze such data. So it's kind of has an educational goal. And so there's nothing here is new research or novel ideas. It's just demonstrating how we can use traditional simple tools to analyze this important data. Okay, and it's based on discussion that I had with Nati Srebro and his wife Ellie. And okay, so let's get going. So, why do I? Okay, so what do we know? So we have in the news, we know results from two experiments, the Pfizer and the Moderna. So how did these experiments go? So the Pfizer, they took 44,000 roughly participants, and half of them were randomly, picked, half of them were given the vaccine, and the other half got a placebo. And nobody knew whether they got a placebo or they got a true vaccine. And then the participants were asked to report symptoms. And if they reported symptoms, so once a week they called the participants and asked them if they have symptoms, and anyone that has symptoms was tested for COVID, if they were symptomatic. And these are the results that we got from Pfizer. So at the first stage, they had 24 participants that were found with COVID. At the end of the trial, they had 170, and 8 of those belonged to the half that received the vaccine. So although initially they administered half of them with vaccine and half of them with placebo, when you look at those that got sick, clearly there were many more from the group that got a placebo, which indicates that the vaccine did something. What happened in the Moderna experiment? So the Moderna was very similar experimental design. So they included 30,000 participants. Again, random half got the vaccine and random half got the placebo. And they were asked to report the symptoms. And here, initially, we got 95 sick patients. Eventually, at the end of the trial, there were 185, and 11 of those are among those that got the vaccine. Again, we see that the big majority of those that got sick were getting the placebo and not the vaccine. So one more thing to bear in mind, here, I hope that you see the bottom of the slide, is that at the same time, about 2,000 experiments with vaccines and treatments are going on around the world. And that's something to bear in mind because many experiments are being conducted in parallel, and we only hear about the most successful of them. Okay, so what would we like to find out? We would like to find out by how much does the vaccine reduce your chances of getting infected with COVID. So the first thing to note is that they only tested symptomatic participants. And therefore, the best that we could hope for is to find how much the vaccine reduces the chance of getting symptomatic COVID, because we don't know how many of them got the virus, but did not show symptoms. But let's just keep it in mind and do the statistical analysis for analyzing how likely is the vaccine to reduce your chance of getting symptomatic COVID. So the first question, the natural question to ask is, can we estimate the chances of getting sick if you get the vaccine compared to your chances of getting sick if you didn't get the vaccine? So let me introduce my first notation. Let P denote the probability of getting symptomatic COVID without the vaccine. So remember, let's say P stands for placebo. And Q, the probability of getting symptomatic COVID when you're vaccinated. So the Q is for vaccinated. Okay, so we have this notation, and we would like to estimate the P and Q. What's your chances of getting sick? So if we try on this data to estimate P and Q, we have experimental results. The question is, how reliable would these estimates be? How much of it could be just due to randomness, to chance, and so on? So a basic tool so we have empirical results, and we are trying to estimate expectations in the population. So the best, I mean, it's a very kind of a mainstream tool for this is to estimate the gap between the true expectation and the empirical average of randomly the outcome of M trials is the Hoeffding inequality. And the Hoeffding inequality tells you that for every epsilon, the probability that the gap, so X is the gap between what we see in the experiment and the true expectation in the population. The probability that this gap is bigger than epsilon is at most e to the minus 2 epsilon square m, so it goes down exponentially fast with m. And A and B are the range of your variable, but here our variable is just the probability of getting sick, so it's between 0 and 1, so we don't have to worry about this A and B. So if you look at this bound, the first thing to note is that it is meaningless. There is no guarantee unless m is greater than 1 over epsilon squared, because here, if m is less than 1 over epsilon squared, then we will get in the exponent more than 1, or more than half. And therefore, telling you the probability that what you see is not reflective of the reality, the probability is bounded by 1. It's meaningless. So we really need to make sure that our sample size is significantly more than 1 over epsilon squared, where epsilon is the control over the gap between the experiment and the true expectations. If we introduce here, substitute the numbers that we have from those experiments, so we want to distinguish between 8 vaccinated people that got sick from the 44,000 and 162 people, I'm talking about the Pfizer experiment, 162 of the 44,000 that got sick without the vaccine. So our estimate for P is this, our estimate for Q is this. The question is this empirical gap significant? So we want to have our test such that the epsilon, the reliability, will be less than the difference between these two. Now, the difference between these two numbers is roughly 1 over 300. So in order to estimate them with enough reliability to say that these two empirical results are really reflecting differences in population expectation, we would need roughly 1 over 300, that's our epsilon squared, so we'll need 90,000 participants. So this is more than we had in the experiment. With the Moderna, if we do the same kind of calculation, we can conclude that in order to estimate P and Q reliably enough that we could tell that they are different, we would need 50,000 participants. So in both cases, the data that we have does not suffice to give us meaningful estimates of the probability of getting sick if you get the vaccine versus the probability of getting sick if you didn't get the vaccine. There is another issue here, is that if we just wanted to know the probability of getting sick with or without the vaccine, we needed to have a sample of participants that reflects the distribution in the population. But those ratios that we have now, they only give us the probabilities of getting sick among the trial volunteers. And this may differ from the probabilities of getting sick in the general population. So if we wanted to use this number to estimate probabilities of getting sick in the population, we would have had to sample completely randomly the participants in the experiment. But that's not the way it was done. They asked people to volunteer for the experiments, and it's likely, very likely, that the volunteers are different than the general population. They are more educated, they're more aware of the risks of COVID, maybe they're taking more precautions. So what I'm saying is that we have two reasons not to use those numbers to try to estimate P and Q. One of them is that the numbers, the precision that we can get is too small with these sample sizes to distinguish between whether this difference is significant or not. And the other issue is that if we wanted to use this experiment to estimate how likely you are to get sick, we cannot trust that the participants in the experiments are really representative of the population. So the story is not ending here. There are things that we can indeed estimate reliably. So instead of trying to estimate P and Q, we are looking for the protection ratio. So protection ratio is a notion that is common in epidemiology, and this is the P minus Q over P. So this is the difference between your probability of getting sick if you get the placebo, to the probability of getting sick if you got the vaccine, divided by the probability of getting sick. Not your probability in the population, but this ratio is the probability of getting sick. So is there a question there? I see that there was a...
Hi, just a quick question. The analysis based on the upper bound and also IID assumption, do you think should be necessarily like reflecting, like the P and Q that we expect?
Right. So what I showed you is this upper bound of the Hofding is not good enough to give you reliable results. We'll see later how we can use better tools than the Hofding. But as a ballpark, it is indicative. I mean, if you want to estimate very small numbers and the difference between them, then these sample sizes would not have sufficed.
Thank you.
Yeah, is there another question?
Yeah, sorry, I may have missed this, but at the beginning, did you say that the two groups were similar on certain characteristics, the two groups that were getting the test?
The two groups that were, what do you mean the two groups that were getting the test?
Well, one that was getting the placebo and one that got the...
Oh, so they randomly, so they flipped an unbiased coin for every participant, whether to give it the vaccine or the placebo. So the partition into these two groups is completely blindly random.
Okay.
So we can estimate that they are similar. Yeah. We can assume that they're similar.
Okay.
So what I'm saying is that let's look at this protection ratio. Maybe this is something that we can do a better job with. So what is this protection ratio? So this is what you see reported in the news. When they tell you that the efficacy of Pfizer is 95% or Moderna is 94%, this is actually where they got the numbers. This is the empirical estimates. They had 160 people that got sick with the placebo. They had 8 people that got sick with the vaccine, divided by the number that got sick with the placebo, it's 0.95. And if we substitute the numbers for Moderna, it's a very similar number. So what you get reported in the media about the efficacy of the vaccine is actually these numbers. These are the empirical results. the question is, what are the true population values? How much can we trust that these empirical results really reflect the probabilities in the true population? So in order to estimate the reliability here, we will use the standard Bayes law, or the Bayes trick. So what is the Bayes trick? The Bayes trick is saying that rather than directly estimating the probability of getting sick given that you are vaccinated, we flip it on its head and say, what is the probability of being vaccinated given that you got sick? So what does it mean? So that's the usual Bayes trick. What does it mean? Say you are a doctor in a hospital and you are being called to see a patient that was, you know that he was part of the experiment and he tested positive. How likely it is that when they will tell you what he got, he actually got the vaccine? So now we're looking at something different. Given that a patient is sick, what is the probability that he got the actual vaccine and not a placebo? The Bayes rule tells me that if I want to estimate the probability of A given B, then it's just the probability of A and B divided by the probability of B. In our case, A is vaccinated, B is sick. So we want the probability of being vaccinated given that you are sick. So this is B is the probability of being vaccinated. Given that you are sick is P plus Q. These are the sick people who got the placebo plus the sick people who got the vaccine. So now what we want to estimate is this number. And again, we have empirical estimates of this ratio, and we want to get the true ratio in the population. So what have we gained? So first of all, note that knowing this V, this Q over P plus Q, readily discloses the protection ratio. Because if the protection ratio, remember, was P minus Q over P. This is your probability of getting sick without the vaccine minus the probability of getting sick with the vaccine as a proportion of the probability of getting sick. So there is a very simple formula that translates between the two. So if we will know this V, we would also be able to estimate the protection ratio. The other thing that we gain, I mean, we can do a sanity check here, say if the protection ratio is 0, the vaccine does nothing. Then if I'm a doctor and I see a patient and I have to estimate, did they get the vaccine or not, my guess will be exactly half, because half got the vaccine and half got the placebo, right? So if the effectiveness is 0, we should get V equals half, and that's what we get here. So if the effectiveness R is 0, we get here 1 minus 0 over 2 minus 0. So just a sanity check that those formulas make sense. What else have we gained? So now this can be thought of as a coin toss. And we can reliably estimate it from empirical data. So what do we have here? So we think of the patient that comes to the hospital with a positive test as a coin, and it's you flip it, its head if it turns up that he got a true vaccine, and its tail if he was not vaccinated. So what we are trying to estimate here, this V, is the probability of heads. This is heads divided by heads plus tails. So this is like estimating the bias of a coin from seeing the result of coin tosses. Is there any question here?
No, there's no question in the chat.
Okay. So, and the other thing to note is that the range of this variable that we're trying to estimate, V, is 0, 1. So we didn't increase the range of the variable, but the values here are much smaller. It's not 8 over 44,000. The values here are going to be the number of sick people, like 8 divided by the number of sick vaccinated divided by the number of sick. This is 8 over 160 or something. We got much larger numbers, and therefore it allows us to draw significant conclusions from weaker approximations based on the empirical results. So we made progress. Now we have variables that are more likely to be reliably estimated because they're smaller ratios. And we also know how to analyze it because it behaves exactly like a coin flip. You see that the results of coin tosses and you try to estimate the bias of the coin. And from that, we will be able to calculate the protective ratio. Okay, so let us try to estimate this. If we try to use, again, the bound, the Hofding bound, so this is kind of the educational part. What tools do we have in order to estimate the reliability of our experimental results? So if we try to do the Hofding again, and we substitute the numbers, so again, I mean, assume what we want to do is do the following. Assume that the protective ratio is less than 0.6. That is, the vaccine is not really effective. Its protective ratio is less than 0.6. And the question is how likely it is that we will see such imbalance between the vaccinated and non-vaccinated among the sick people just by chance, just by random. So that's what we want to estimate. So if we assume that the protective ratio is less than 0.6, we get that V is 0.285, and we substitute the number, and we get that the gap that we need to estimate is 0.23. The epsilon now is much bigger. So that's the gap between the vaccinated, the empirical result and the theoretical result that would get if we assume that the vaccine is not effective enough. So the gap that we want to estimate now is 0.23. And if we substitute it in the Hofding, we get that the probability that we'll see such a result by chance, if the vaccine is not effective, is roughly 1/7,500. So it looks like very unlikely that we will see it by chance if the vaccine is not at least 0.6 effective. Still, it's not the end of the story. Why is it not the end of the story? I mean, on first sight, it seems pretty reassuring. It's highly unlikely, probability 1/7,500, that the vaccine is not protecting you enough, and yet we will see such results by random. But you have to remember that we have about 2,000 clinical trials running at the same time. And we only hear about the most successful. So you can think of it, as if you have 2,000 coins, and each of them is being flipped, I don't know, 100 times. But I report to you the coin that got the highest number of heads. Even if all the coins are the same, the fact that one of them got high number of heads doesn't mean that they are not biased. So even if each of those experiments is completely useless and they have success probability of below 1/1,000, if you have 2,000 of them running in parallel, the probability that at least one of them will give you such results as what we've seen is highly significant. So we have to somehow take into account the fact that we only hear about successes and we don't hear about all the failures. So how do we do that? The most naive way, I mean, I will be happy to discuss more sophisticated ways later at the end. But the most simple way of counting for that is the Bonferroni correction. So let me just remind you, the p-values is what we've been discussing so far. It's the probability of seeing test results as we saw, given that our assumption, the null hypothesis, that the vaccine is ineffective. So our null hypothesis is that protection factor of vaccine is below some value R, or 0.6. And the p-value is the probability that under such an assumption, we'll see the nice results that we saw. If there was only one experiment, we saw that this p-value was 1/7,500. The Bonferroni correction says if you have you want to protect against cherry picking the most extreme case among all the trials. So it just says multiply your p-values by the number of parallel experiments. So here we will multiply our 1 over 7,500 by 2,000, and we'll get something like 0.27. So this is not very convincing. It means that because so many trials are running in parallel, the chance that at least one of them will give us so significant results is 0.3. That's not very reliable. Still, we can do better. Why? Because what we did so far is we used the bound from the Hofting that was referring to the question that I was asked before. The bound for the Hofting is very, very loose bound. We can get better bounds. So one way of doing a better job at estimating the probability of seeing such results by chance is the Gaussian approximation. Since now we are in a situation where we are just tossing a coin, we know that the distribution of the results of coin tosses, as the number of coin tosses go to infinity, it gets closer and closer to a normal distribution. And we can calculate the mean, we can calculate the variance, and we have tables that tell us what is the tail probability. If the mean is here, what's the probability that we'll see a result that is very far from the mean? So you can just, I encourage as an exercise, put these numbers into estimation of the variance and of a Gaussian and see how likely it is. We get better results than the Hofting bound. What I have here is something even better. So that's, Nati helped me. He ran a Python program to calculate those probabilities exactly with these numbers. It's not such big numbers that you cannot run the exact calculation. And this is what you get for the exact calculation. So let me explain to you what this graph shows. So what we have here is the protection ratio. So this is assuming that the protection ratio is less than this number. What's the probability, the p-value, what's the probability that we will see such results by chance? And the blue line is if we view this as the only experiment running. And the green line is after the Bonferroni correction, bearing in mind that there are many experiments and we only see the most successful. And what we see here is that if we draw the line of reliability at 0.05, at p-value 0.05, there is, we are trusting the results if the chance of seeing such results by chance is below 1 over 20. Then we see that we can trust that the protection ratio of the vaccine is almost 0.65. Now, for government authorities to authorize, to confirm a vaccine, it's once the vaccine effectiveness is above 0.5. So here we see that with pretty much confidence, we can say it is above 0.5. And so we can trust that the vaccine is reliable and definitely above 0.5, because if it was only 0.6 effective, the likelihood of seeing such a results would be like 1 over 200. And if it was only 0.5 effective, it's completely negligible. So definitely the vaccine is effective. Okay. And another thing that you see in the news is the report of severe symptoms. People can say, it's not so bad to get COVID. What I really worry about is getting COVID with severe symptoms. So we can repeat the same kind of analysis on the severe cases. So in the Pfizer trial, there were 10 participants that became seriously ill, and all but one of them were in the placebo group. So if we just look at severe cases, what we see is 10 severe cases showed up in the Pfizer experiments, and only one of them got the vaccine. Now we can repeat the same analysis. We can say how confident can we be about the efficacy of the vaccine with respect to severe symptoms. But note that now, although the ratio looks very extreme, now our denominator, the number of patients that came to the hospital with severe symptoms were only 10. So the sample size that we are dealing with is much smaller. So in this case, V, the probability of seeing someone who got the vaccine among the severe symptoms cases is 1/10. And if we even assume that the vaccine is only 0.6, is less than 0.6 protective, we'll still have a probability of more than 0.10 to get such results. So here I drew this graph. This is easy to calculate. You can see that if we want protection against severe symptoms, and you want to put your p-value at 0.05, then the protection ratio that you can reliably assume is pretty low. And this is even before we take into account the repetitions of many experiments. So the conclusion is that although it's very impressive, there is not sufficient evidence to conclude high efficiency in preventing severe cases. Not because it doesn't prevent them, just because we rely on how many such cases happened, and too few of them happen to get statistical reliability with respect to severe cases. And the results with respect to severe cases with Moderna are stronger, they're more significant, but still, we have to bear in mind that we have two things that we can try to estimate. We can estimate the proportion among the full population of being sick or being severely sick or being severely sick with vaccine and so on. And there we have problems because those numbers are so small that we need sample sizes bigger than what we have in order to estimate them to distinguish between the vaccine and not vaccine. The other option that we have is to look at the ratio of vaccinated among those that got sick or among those that got severely sick. But then our sample size is limited. It's how many sick people did we see in the experiment, and therefore it hurts our reliability. So let me move on to the take-home points. So first of all, we have strong evidence to support the claim that these vaccines are overall effective. That means we can trust that they reduce the chance of symptomatic COVID. And that's very good news. However, we have a few caveats. So first of all, as I said, we would like to approximate P and Q, the probability of getting sick. We don't have enough data for that, and the experiment was not designed for that because we didn't pick random people from the population. The other point is that, so instead, all we can estimate is the probability of having received the vaccination, given that you are sick, but you have to bear in mind that we only see the empirical results, and they may be different than the true population probabilities. Although in the news, what you see is the empirical results as if they are the probabilities of success in the true population. What are the statistical caveats? So, of course, are the trial participants a faithful representation of the target population? And the reliability of estimating the true V from the empirical V, it depends on something that we don't have control over. No one has control over how many sick patients we will see, or how many severely sick patients we will see, or how many sick people we will see among a certain age group. And since we don't have control over this, our reliability really depends on, by chance, how many happen to be in this group. So when it comes to subpopulations, these numbers are often not publicized. They're not telling us exactly how many sick among each of the groups. and the numbers are becoming so small that we cannot infer significant statistical significance, like we saw with the case of severe symptoms. Furthermore, what we see now is that people are trying to evaluate many random variables from the same single experiment. They want to know efficacy by age group, efficacy by gender, is it more effective for men or for women, efficacy by ethnicity, the risk of different side effects, the time to onset. And if you're trying to estimate many random variables on the same data, the statistical validity degrades because we are increasing the chance that at least one of those things that we are estimating will be there just by chance. So I would be very skeptical about any attempts to say, oh, in this group, we saw only one patient under the age of 30 that got sick with the vaccine. It is not necessarily significant because we are asking many questions on the same small data, and we cry out only those that turn out to look significant. So for subpopulations or for any kind of other random variables that the experiment was not initially designed to estimate, it's not a good idea to trust such results. On top of it, there are many clinical issues. I mean, we were only symptomatic participants were tested. Maybe the vaccine suppresses symptoms, but people can still be infectious. We have results at a given point in time. Maybe the vaccine has certain lengths of time in which it affects and then it degrades. It's unclear the relationship between the efficacy and the group of patients, the risk of allergic reactions, we hear about it more. So still many unknowns, clinical unknowns. From the statistical point of view, we can trust that the vaccine is effective in reducing symptoms of COVID. Any discussions about subpopulations are not completely reliable. So that's the story for today. And I'm happy to take questions.
Thank you, Shai. Yeah, does anyone have any questions? And if you don't want to put your microphone on, then feel free to type them in the chat and I can forward them on to Shai. Oh, so not a question about your talk, Shai, but one person is asking if you would be willing to share the slides.
Yeah, definitely. I mean, I can send the slides to you, and you're welcome to share them, and also the video of the.
Yes, of course.
Yeah, definitely. And if anybody has questions later, I'll be happy to discuss it. I think it also interesting to me. Sometimes people come with very, like, for example, one of the questions I was asked when I presented this talk earlier was, say, Moderna and Pfizer are not just any random startup that's trying to develop vaccine. Why do we think that they're all same likely, and give each of them the same weight? And this is a very good point. And we can do a more sophisticated analysis of how likely it is that one of the big companies will succeed. But we need to assign weights to the different companies in advance, not after we see the data. But we can do it, but we have to say this company is important. If they show results, it's more significant than that company. But we have to assign those weights before seeing the data in order to conclude that the weighting was significant. If we didn't do it in advance, the best that we could do is this, giving the same weight to any of them and doing the times 2,000 reduction of the confidence.
Yeah, that makes sense. Okay, well, if there's no more questions, then we can wrap today's talk up here. Thank you, Shai. Oh, Sorry, no, that was someone sending me a message. I thought it was a question. Sorry. As the feedback is saying in the chat, your talk was very informative and very well explained. So thank you so much for your time today, Shai. And thank you to everyone else for coming. As I said, this is our last Thursday seminar for the year. So happy holidays to everyone. And we'll be back in 2021.
Okay. Nice holidays. Bye.
Yeah, you too, Shai. Thank you. Bye, everyone.